Well, good evening and welcome to the next installment of the SLAC Public Lectures. And thank you all for being here. Our speaker today is Taran Driver, who's a postdoctoral fellow here at SLAC. Taran, I guess, grew up in England. He did his PhD work at Imperial College in London, then came here for a postdoctoral appointment. He's been doing some very cool stuff. I guess that was recognized this year by the award of the 2023 LCLS Young Investigator Award. So congratulations. And at the same time, over on the other side of his abilities, he won our local physics slam competition, where you have to explain yourself in 3 minutes with basically no visual aids. And for this, he won a trip to Washington. But I think there he just has to explain to our congressman all the cool things we do. With congressmen, you only get a couple minutes. Here, you've got him for a whole lecture. So let's enjoy it. He's going to talk about, please excuse me, what is the title?
Electrons.
Catching up to electrons on the move. So good luck.
Thank you. Hello, everybody. Sounds good? Amazing. Okay. So yes, as Michael said, my name's Taran. I'm a staff scientist here at SLAC, and I'm going to talk to you today about the work that I've done over the past few years here at the lab on watching electrons move. And I'm going to explain to you what that took. It wasn't particularly easy, and why we were interested in doing that in the first place. We'll start by situating you, though, because I would imagine not everybody here has been to SLAC before. Where we are is in this little red square I've drawn in this aerial view here. And you can make out in this aerial view on the left-hand side of this slide a very long straight line that shoots off into the San Francisco Bay, which is what you can see in the distance. And here we're on the main campus of SLAC. Zooming in, we are in building 51, which is in red up here. And I spend most of my time in building 950, which is over on the other side of campus, but on this campus. And I spend it about 3 floors underground, 3 stories underground in this lab that I've taken a picture of here. And there is some fancy equipment here, and I will explain a little about how I use this fancy equipment later on. And so why is anybody here in the first place? Why have we ended up at SLAC? What brought people here in the first place? Well, SLAC is formerly used to stand for Stanford Linear Accelerator Center. It's now just SLAC. It's become a name into itself. And it is a lab that was built in the 1960s. And it was originally built as a particle accelerator. And the reason there is such a long straight line that you can see in this aerial view is because this line was originally built as essentially an electron racetrack. It was to make electrons go as fast as we could make them go, which turned out to be very, 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 very close to the speed of light, extraordinarily quick. And this is the aerial view that you can see. But if I had X-ray goggles and could peer deeper underground, I'd see what I've shown in this picture here on the left-hand side, which is a series of very long pieces of scientific kits. And these are accelerator modules which cause electrons that we produce at the very start of the beamline over in the bottom left to go quite so fast. And this fella here on the top right is called Wolfgang Panofsky, and it was his original idea. He was a professor at Stanford, and it was his original idea to build SLAC in the first place. And around about the time that SLAC first came online, somebody asked him, how long is the lab going to live? Because, you know, this was a very large investment, and they're interested in knowing if it would have longevity. A spoiler alert, given we're all here, is it lasted quite a long time. But his original answer was, SLAC will live about 10 to 15 years unless somebody has a good idea. Because the lab was originally built to do one class of experiment, which was to peer inside the internal structure of a proton. But subsequently, it has been refurbished and reimagined as many other different pieces of equipment as well. So hold on to that quote, unless somebody has a good idea, because the work that I'm going to speak about is about one of those more recent good ideas. And then Michael did a nice and very flattering job of introducing me. Thank you, but I'll briefly cover my background. You will have guessed, if Michael didn't tell you by the way I speak, that I'm not from here originally. I grew up in the UK. I did my undergraduate dual major, joint honours in physics and philosophy. And that was at the University of Nottingham, Nottingham is a small city in the middle of England, probably most well known because it's near Sherwood Forest. Sherwood Forest is where Robin Hood and his merry gang of men used to run around. Then I moved down to the capital, to London, to do my PhD, where I was interested in analyzing protein structure. And when I finished my PhD, I hopped on a plane, flew out several thousand miles west, and found myself here at SLAC. And at SLAC, I split my time roughly half and half between the lab that I've already shown you a picture of. This is me at least pretending to do some work on a spectrometer in the lab, and sat on computers staring at data, trying to analyze the numbers that come out of experiments and figure out what scientific insight they give us. Okay, so now I'm going to try and tell you about the work I do and why it's important. And we'll begin with a very short crash course on atomic and molecular physics. I'm sure these pictures will be somewhat familiar to most of you, depending on how long ago you studied physics and chemistry. On the left here, I have a basic model of an atom. And you can think of an atom as consisting of two parts: a nucleus, which is made of protons and neutrons, words you're probably familiar with. These are small charged particles that form the nucleus, the center of the atom. And then around the outside, you have these electrons, which move around in some fashion on the outside of the atom. And I can join a bunch of atoms together to form a molecule. And one way of drawing a molecule, which is just a collection of atoms joined together, is this ball and stick diagram that I have on the right. So here, in this picture here, the balls correspond to the nuclei. The balls that I have here, each of those correspond to the nucleus of a particular atom. And the electrons are drawn slightly more abstractly, but all these sticks you can see, which are chemical bonds, are formed by electrons. So the electrons themselves form these sticks, which join the different atoms together, join the different nuclei together. And there's a reason I chose this molecule here. Well, there's a couple of reasons. One of them is the work I'm going to speak about was done studying this molecule. Another one is, and this is, I guess, the reason that we chose to study this molecule in the first place, it turns out to be a very useful prototypical system for us to study. And so I've redrawn the molecule here in the bottom left. And the reason for that is this molecule, which is called aminophenol, bears a lot of similarities to molecules which are very useful and very important to us. So say, for example, I have a series of different molecules here. This one directly above the aminophenol is something our body uses to produce DNA. The middle is a molecule known as phenylalanine. This is an amino acid, so it's one of the 20 constituents of all the proteins in our body. This one on the top right, you may be more familiar with as what I call paracetamol, and what you would call something else, which I've forgotten, a painkiller. Acetaminophen, I think that's what you call it, yeah. And you can see these actually bear a stark resemblance. This dashed hexagonal feature of these 6 black atoms, black encodes carbon in this case, is replicated in all of these different structures. And then here, this structure I have on the bottom right, I knew I'd forget the name, so I wrote it down, is sodium picromate. Sodium picromate is used by very many of us as both a hair dye and rocket fuel. So if we're going to use it for both of those, we certainly want to understand how it works and what its molecular physics is. And I will come back to this, but the black atoms that you see in my molecule here are carbon. The blue is nitrogen, the red is oxygen. So the different colors just encode for different types of atoms. Okay. So in order to explain how molecules move, I often like to appeal to an analogy that I will give to you today. So I said that this molecules like this structure here consist of nuclei, which are these balls, and are held together or bound together by electrons. And if we zoom down into the molecular world, it turns out that the nuclei are actually quite big and heavy and move relatively slow. So in my analogy here, the nuclei are going to be the sheep, and the electrons, which are smaller, faster, and zippy and move around, are going to be the sheep dogs. Okay, so I'm going to paint these nuclei onto my these sheep onto my molecule, and it turns out that when nuclei move, so when sheep move, what we all know these things as, so the sheep moving is chemistry. When nuclei move, that's chemistry happening. They're the same thing. Nuclei moving around, breaking and reforming bonds is chemistry happening. And why is chemistry happening? Why are these nuclei moving in the first place? Well, true to the analogy, it's because the electrons told them to. Electrons herd these molecular sheep and make them perform chemistry. And electrons are very fundamentally important things and carry a huge number of different important tasks in our everyday life. So say, for example, this is a circuit board I have on the top left. Well, the things that carry information in that circuit board are electrons. In solar cells that I have at the bottom here, the particles that take solar energy and convert it into electrical energy, of course, you can guess where that word came from, are electrons. And when chemistry is happening, well, the reason it's happening, like I just said, is that nuclei are moving. Why are the nuclei moving? Well, it's because the electrons are herding them and telling them to do so. And so to carry this analogy one step further, why does a chemical reaction happen in the first place? Why would a chemical reaction start in the first place? Well, it's because the electrons, the sheepdog, are moving around and telling the nuclei to move. Why do the sheepdogs move? Well, it's because we tell them to. We whistle at them. And in the same way that a sheepdog might hear a particular sound wave from a whistle and know to do something, our electrons will receive information from light waves instead. So dogs listen to sound waves, electrons listen to light waves. They receive information in those light waves and they carry that information and make chemistry happen. And what I've drawn here for a light wave is something that we use a huge number of here at the lab. It's a laser. But it also happens, as I'm sure you're aware, that there are lots of other light waves in the world. For example, the sun, which produce light all the time, sends signals to electrons to initiate chemistry all the time and cause things to happen a huge number of different places. And we'd love to understand how that is happening, how the light waves are causing chemistry to happen. So drilling down a little further into what especially I'm interested in doing, I like to study photochemistry. So chemistry, you know that word, photo, initiated by light. Photochemistry is chemistry initiated by light. And it turns out when light interacts with something, it makes things happen. So at the top, I have a leaf. When a light shines on a leaf, the leaf can generate energy, which then feeds all living matter. If sunlight shines onto a DNA molecule in the top right, it can cause bonds to break. If things go wrong and this perpetuates, ultimately it can lead to sinister diseases like cancer. The photochemistry is also actually the reason for which champagne bottles end up being green, or at least why they were originally made green. It's because the green glass protects the champagne from sunlight. It stops the photochemistry being initiated by the sun and stops the champagne tasting off, or at least giving you a headache. So the question that remains unanswered about this photochemistry is we know that light can shine on something and make things happen. We know that the electrons carry the information. They receive information from light and they cause chemistry to happen. What we don't know really anything about is any of this part that I've labeled in red. We know that there's a start product. We know there's an end product. We know the electrons are doing something, but we really don't know what they do. For example, we don't know which whistles, which flashes of light cause which chemical reactions. We don't know what the most efficient way to initiate a chemical reaction is. And we also don't really know how the electrons bark at the sheep. We don't know how the electrons talk to the nuclei, although we know that they do exert some impact. So put very simply, the task that I want to do, the task that I'm seeking to do, is to catch electrons in action. Here is one final analogy. Here I am, sat with my high-speed camera on the side of, I think this is a soccer pitch, and there are the electrons. These are these fast, zippy things moving around. And the aim of what I'm doing is to sit and take high-resolution pictures of electrons moving, and in doing so, understand what they're doing, understand how it is that electrons precisely are talking to the nuclei and making chemistry happen. But it's not particularly straightforward, of course. There are a few different things that make it hard to do this, and I'm going to talk about three of the most tricky things to do with capturing electrons. One is, as you may have guessed, electrons are very small. Two is, they behave according to the spooky laws of quantum mechanics. And three, which was in the title of my talk, so you can guess this was important, electrons travel really, really, really fast. Okay, so I'll run through each of these different challenges. Electrons are small, they're quantum mechanical, and they move very fast in turn, and try and help describe a little about how we can overcome these challenges to take pictures of electrons. Okay, so to give you a description of why electrons are small, I have two props. I guess how small electrons are. And maybe somebody, can one of you tell me what this first prop is? It's small, it's green. Yes, and what is it? What am I holding? It's a, pretty much, it's a P. It's a P, exactly. So I'm holding a P. Good job. And I'm going to take you to a world where I have the ability to take this P with all its constituent molecules and blow it up really big, really, really, really big, in fact, to the size of the Earth. And this P has loads of molecules in it. So if I blew this P up to the size of the Earth, my question is, and this pertains to the next prop I have, well, how big would a molecule in that P be? So I have my one P, everything in the world stays exactly the same size, except this P. I blow this P up to the size of the Earth. How big are the molecules in this P? Does anybody have an idea? Would you like to guess? Sorry? Yes, my second prop is also a P, correct. Great job, exactly. If I blew a P up to the size of the Earth, well, the molecules in that P would end up being the size of a P. And that's a molecule I'm talking about. I said the electrons correspond to the sticks inside that molecule. The precise size of an electron is not particularly well defined, but it is a fraction, a small fraction of the size of a molecule. So here you can see how molecules are really, really very small. Electrons are slightly smaller. And that presents a problem, because if I want to take a picture of something, a picture of an electron, or indeed a picture of anything, I need the wavelength. Maybe, have I got it illustrated here? No. The wavelength is the distance between two consecutive crests or two consecutive troughs of light, which is a wave. I need the wavelength to be comparable in size to the size of the thing I'm trying to take a picture of. So say, for example, down here on the bottom left, I have a picture of the face of a fly. And you can see its big, gruesome eye there. The big eye is about 100 microns. A micron is 1/1,000,000th of a meter large. And I can take a picture of a fly with visible light. Visible just means the region of the electromagnetic spectrum that my eye is sensitive to. And if you peer a bit closer, especially in the front, you might be able to see that the eye is actually divided up into a bunch of different subunits. These have a name that I also knew I'd forget. Ommatidia. And these are tiny little fractions of an eye. And these ommatidia are about 1 micron large. So they're much smaller than the full eye. And in reality, when we start taking smaller and smaller pictures, the ommatidia are about as small a thing as we can take a picture of with visible light, with light from the sun or light that comes into our eyes. If we want to take a picture of an electron in a molecule, we need the wavelength to be about 1,000 times smaller. And this takes us into the X-ray regime of the electromagnetic spectrum, where we have a wavelength of about 1 nanometer, 1,000 times smaller than a micron. And a problem here is that visible light is available to us everywhere. It comes from LEDs, it comes from the sun, it's in abundance. X-ray light is much harder for us to generate and presents a technical challenge for us to be able to take a picture of a molecule. I wanted to very briefly explain a little more about why the wavelength needs to be the right size to look at the particular object. And the analogy I'm going to use is my hand. Okay, so say my hand corresponds to light. I'm trying to figure out the shape of something with my hand. Well, if I close my eyes and rub my hand across a fence that has this crisscross pattern in it, well, my hand's about the same size as one of these units. I can feel where there's no wire, I can feel where there's wire, and understand the shape a little bit. I know, though, as well, that the same pattern appears in the material on my shirt. But if I shut my eyes and run my finger over my shirt, even though I know there's this crisscross pattern there, well, my big old finger is way too big to be able to feel where there's cotton and where there's no cotton. And I can't resolve with my big fat finger these much smaller holes in a shirt. So in the same way, we need much smaller wavelengths than visible light to take pictures of molecules. And you'll remember this man on the top right saying, SLAC will live for 10 to 15 years unless somebody has a good idea. Well, one of those good ideas was first conceived, I don't know, several decades ago, came to fruition in 2010, and it is the Linac Coherent Light Source, or the LCLS. And this is a refurbishment, a reclaiming of the particle accelerator that makes electrons go fast. Instead of making electrons go very fast and slam into something, which was how the first experiments were designed here, the LCLS takes these very, very fast electrons, passes them through a series of alternating magnets. So what you can see here in red and green is magnets. Red is north, green is south. They flip around. And when the electron goes through these magnets, it's made to wiggle. When an electron wiggles, it produces a pulse of light. And because the electrons are going so fast, the color of light, the wavelength of the light that's produced is X-rays. So the LCLS is actually the brightest, right here on this campus, is the brightest source of X-ray pulses in the world. You cannot get brighter X-rays anywhere. So it's a good place to study molecular physics and to understand how electrons move around molecules. Okay, so first challenge, electrons are really small, but happily we have an X-ray laser. Okay. The next problem I mentioned is electrons behave, I'm going to take a sip of water, according to the funky, bizarre laws of quantum mechanics. And this presents a conceptual challenge to everybody, including those who think about these things every day. And it turns out that one of the main fundamental challenges and logical inconsistencies with quantum mechanics is that an electron can exist as a wave and as a particle. I can do several experiments on an electron, and I see it behaves like a particle. It does everything I expect a particle to do. It exists in one place, for example. I can then do a bunch of other experiments on an electron and see that actually it looks more like it behaves like a wave. And this throws up a whole bunch of questions. For example, if I take the X-ray laser I just spoke about, and I look for an electron on my aminophenol molecule, I can very clearly see it is on one end of the molecule and not on the other end. Then I do several different experiments. I see the electron behaves like a wave, but I know that a wave exists throughout space. And how can I match those two things? How can the fact that I see an electron in one place and not another place be consistent with the fact that I know an electron behaves like a wave, which is present through all space? Well, it's a challenge. I will try to dig slightly into wave mechanics, into the wave formulation of quantum mechanics, to explain how we can square these two things. And I will do so, I'll begin doing so by discussing more a wave that probably many of you are familiar with, a water wave. Okay, so how do we interpret what a water wave says? I have a wave of water which is drawn here, and I can see a peak in the water wave and a trough in the water wave. What does it mean when I see a peak? When I see a peak, I know that beneath that peak, there is more water in my lake or in my bath or whatever. And when I see a trough in my wave, I know that equally beneath that trough, there is less water in my lake. So a peak means more water, a trough means less water in a water wave. It's not that different from how we can interpret an electron wave, a wave that describes an electron in some bizarre way. If I see a peak in my electron wave, it means that the electron which this wave describes, I have one wave, which is this purple thing, it's describing one electron. A peak in my electron wave means my electron that I'm describing is more likely to be there. That's on the left-hand side. A trough means the electron I'm describing is less likely to be there. Peak, electron more likely. Trough, electron less likely to be there. And I said at the start that an electron is a wave. It may have been more straightforward. It may have been cleaner to say an electron is in fact a combination of waves. I don't just need one wave to describe an electron. I need a bunch of different waves to describe an electron. And we can appeal to something that probably everyone is familiar with to explain why this is necessary. So here in this video, you have two little bobbers which are making two waves, two individual waves. And in the middle, as these waves in this lake meet, they are crisscrossing and overlapping and forming an interesting pattern. This is an interference pattern. We can run over it again. And you see this fairly simple shape made by one bobber is being made into something much more complex by the introduction of additional bobber and two different waves there. So that what the streak down the middle is a much more complex shape than the two waves individually. And the law for how waves combine turns out to be extremely straightforward. It's the same for electron waves and for water waves. It's essentially the same for the two types of waves. And the way in which two waves combine, so here, I've called these wave components. Think of this as being one bobber and another bobber. I have two bobbers which are making my waves in the water. These combine to form my total wave. One purple total wave is still describing one electron. It's just made of these individual components. And the way I describe my total wave is just the sum of the two wave components. So say, for example, here at position 1, I have a negative in my first wave component, a negative in my second. I sum those numbers together, I get a bigger negative in my total wave. Equally on position number 2, I have a positive, another positive. I sum those together, I get a larger positive. We can ask what happens if I shift this second wave a little bit to the right. Well, here's something interesting happens. At position number 1, I have a negative and then a positive. At position number 2, I have a positive and a negative, and they all sum to give 0. So my total wave is the sum of the wave components of the two different components. Okay, so I will say once more, my purple wave is describing one electron, one wave describing one electron. And I'm making that wave from a bunch of different components, which are themselves waves, that I sum to get my total wave. But how does that help us explain this funny thing, which is that this green spot on the left-hand side of my molecule, which is an electron, appears at one place. When I look at it, I see an electron at one place, not distributed throughout space. Well, let's build up a total electron wave through a series of different electron wave components. And I want you to keep an eye on two places on this total electron wave. One is this black region, where we wouldn't really expect to see any intensity in our total electron wave because the electron isn't there. And the other is this red region, where we do expect to see intensity because when we do an experiment, we see the electron at that place. Okay, so we're going to build up our total electron wave with a series of quite carefully chosen components. Okay, I sum the first one. I have on the left-hand side, the red, positive plus positive makes something higher. On the right-hand side, my two green components, one is positive, one's about 0. It sums to something smaller. I can add another wave and another wave. And it turns out that by carefully choosing the electron wave components, my total electron wave that describes a single component can be made to have a really quite high peak where I see my classical particle, where I see my electron, and be pretty much 0 elsewhere. So I can actually describe many of the properties of a particle just by using waves with wave mechanics. Okay, so that's great, but that's a stationary electron. It's an electron that doesn't move. What about thinking about motion? So the difference between a particle and a wave in terms of motion. Well, a particle can move from one side of the molecule to another. We know that. But a wave doesn't have the same motion. It sort of wobbles up and down. How can we square these two different types of motion to see that there's a comparison, there's not a logical inconsistency between an electron being a wave and a particle? Okay, well, I'm going to decompose my total electron wave into a series of components again, but this time I'm going to let each of my electron wave components move like a wave. How does a wave move? We just saw it moves up and down. Okay, and then I go add my second wave component, and that's moving up and down. It's moving slightly faster. My third wave component is moving slightly faster still. My fourth wave component is moving even faster. So now the mental task I would like you to do, which is going to be impossible, is to sum all of these waves together and tell me what the total electron wave is doing. I'll give you a couple of moments to compute. That's right, you got it. It turns out that if I choose my waves and I have them oscillating at all different frequencies, well, the total electron wave gives me motion that looks very, very much like a particle's motion. It has a peak that moves from one side to the other, back to the other, back to the other. A little bit like a mouse running under a rug. And this is how I can square my wave picture of quantum mechanics, where I know that electrons behave like waves, with my particle picture, where I see particle-like characteristics of an electron when I measure it. Okay, great. So Second problem down. Electrons behave according to quantum mechanical laws. They are very funny things, but at least we all now in this room understand the basics of the wave mechanics formulation of quantum physics. Cool. Okay. I will move on to the last one. The last problem I spoke about. And this is quite big and has been something that has taken us really, well, several decades to fully get our heads around. Not me, myself, but colleagues. Okay. Electrons move really fast, really, really, really fast. I'm going to try and explain to you quite how fast electrons move. Here is my little electron dog, and it's crossing a chemical bond. The time it takes an electron to cross the chemical bond is about 100 attoseconds. I don't know how many of you know what an attosecond is, but I will explain it to you. And at this stage, I hope for a small amount of audience participation. We will slice up a second. Okay, so I think most of you know that when scientists talk about things which are very big or very small, we put a prefix in front of a word. A gram becomes a kilogram, or a second becomes a attosecond, for example. So we're going to slice up a second and see how much we have to slice a second up until we get to an attosecond. Okay, so time to shout out, as long as you're not too shy. If I divide a second by 1,000, what do I get? This one's easy. A millisecond, exactly. I'm now going to divide a millisecond by 1,000. Microsecond, yes, bingo. What if I divide a microsecond? Yes, nanosecond, great job. See you all the way. Okay, I see you might be being fed answers here, but that's all good. Okay, we're nearly there. I'm now going to divide a picosecond by 1,000. What was that? Femtosecond, yes, amazing. All right, and now this one's easy. I'm going to divide a femtosecond by 1,000 and get an attosecond, exactly. So what have I had to do to a second? I've had to divide a second up by 1,000, 1,000 again, 1,000 again, 1,000 again, 6 times in order to get to an attosecond. Okay, what does that mean? All right, that's a lot of slicing. We only have human brains. How do we rationalize what that means? Well, here is one way. An attosecond is what I've just described. There are 5, this number is big. This number is 500 quadrillion attoseconds in one heartbeat. This is something that you all know. Heartbeat's roughly half a second. 500 quadrillion attoseconds in one heartbeat. What period of time has the same number of heartbeats in it? There are 500 quadrillion attoseconds in one heartbeat. 500 quadrillion heartbeats corresponds to which period of time? Again, I need some audience participation here. You can just do this by show of hands. Your options are, so you can think about them, a year, a calendar year, the time since the Roman Empire, which I think about daily. Homo sapiens, the time since Homo sapiens was first emerged as a species. And this one here is the Big Bang, or the time since the Big Bang, the time since the start of the universe. Okay, so there are 500 quadrillion heartbeats in a year. It wouldn't be a year, too easy. Okay, there's only about 63 million heartbeats in a year. Time since the Roman Empire. Okay, very good. Still not, 126 billion heartbeats. Time since Homo sapiens emerged. Nobody. Ah, 1, okay. I'm sorry, I don't mean to call you out because you are wrong. There are 500 quadrillion heartbeats in the time since the Big Bang. Since the beginning of time itself, there has been 500 quadrillion heartbeats, and there are 500 quadrillion attoseconds in one heartbeat. Okay? There are the same number of attoseconds in one heartbeat as there have been heartbeats since the beginning of time itself, more than 30 billion years ago. Unimaginably fast. We cannot begin to wrap our heads around that. Okay. So one last piece of audience participation, please. This is going to be an open floor. I told you that electrons cross a chemical bond in 100 attoseconds, okay? You now all completely understand what an attosecond is. So if I said to you, imagine a world where I could take a step every 100 attoseconds, the time it takes to cross a chemical bond, then in the blink of one eye, how far could I run? 100 miles. Close? Any increase on that? It's a good guess. Anyone else? Across the universe. Yeah, okay, that's a great question. A great one. Slightly lower. I'm going to tell you how far. You highballed me. If I could take a step every 100 attoseconds and the time it takes for an electron to cross a chemical bond, then in the blink of an eye, I could run to the sun, and then I could run back, and I could do that 3,000 times. Not quite to the edge of the universe, still quite far. Okay, and this presents a problem, okay? Because we said we're sat on this soccer field, I'm the sports photographer, and I want to take a picture of electrons moving fast. And anyone who's taken a picture of children or animals moving fast knows that to take a sharp picture of something moving fast, you need a short shutter speed, okay? And the shortest camera shutter that it's possible for us to make goes for about 100 microseconds. It's quick. It's like 1 trillion times too slow for us to take a picture of an electron. So we need another idea. We need to do something else. And most of you will recognize the first 5 letters in this word, stroboscopic imaging. It corresponds to a strobe light at a disco. Okay, so what happens in a strobe light at a disco? Well, it flashes on and off very, very fast. It produces short pulses of light. And if you keep your eye open all the time, so you're not changing how long your camera shutter is open for, you get to freeze the dance moves of someone in motion. You see them moving, you see nothing. You see a frozen shot of them moving again. You see nothing. You see a frozen shot of them moving once more, and you see nothing. And this idea of stroboscopic imaging, keeping the camera shutter open all the time and flashing a system with very, very short pulses of light, has actually enabled us to take pictures of things moving extraordinarily fast. This is one example. This was done in the 70s. And this is a picture of a bullet just passing through an apple, taken with a flash of light that is 300 nanoseconds long. Here, the camera shutter has basically been kept open for forever. And the flash of light captures the bullet still. You can see the etch on the side of the bullet, and you can see the apple exploding. So 300 nanoseconds is very, very fast, of course, but you might remember from our slicing exercise, we are many, many, many factors off an attosecond. And so if you had heard of the word attosecond before, it might have been because this is quite timely. The Nobel Prize in Physics this year was awarded to Pierre Agostini at Ohio State, Ferenc Krauss in Munich, and Anne Willier in Lund for being the first physicists, the first anybody, to generate pulses of light that last for just a few attoseconds, incredibly short pulses of light. And the reason that was important is the reason that I'm going to describe today. It's because we now can do stroboscopic imaging with these attosecond pulses of light on molecules. And so my strobe light is the X-ray laser I discussed at the start. Not only is it the brightest source of X-rays in the world, it also produces some of the shortest duration X-ray pulses. And my talk isn't going to go into the details of how you make these attosecond pulses. You can trust me that it is an extraordinarily complex technical development. Happily, Siqi Li gave a public lecture in September that describes the way in which these attosecond pulses are made. You can watch it on YouTube, and I will call out that these two people here, Argo Marinelli and James Cryan, were really the driving people behind generating these ultra-short attosecond flashes of light with the X-ray laser. So it says on the top, my strobe light is the X-ray laser, and that is what we use to look at electrons in motion. And a molecule. Okay, so coming back to my analogy, here I am. I should be looking slightly happier now because we figured out how to take snaps of electrons. These electrons are moving fast, and we've overcome these problems that we spoke about. Electrons are really small, but we have an X-ray laser. Electrons behave according to the laws of quantum mechanics, but now we have an understanding of wave mechanics, and electrons move really, 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 really fast, but we have a strobe light, we have an X-ray laser that makes attosecond pulses of light. So the last thing I'm going to speak about is bringing these things all together and telling you about how we made a measurement of a dog moving across a molecule, an electron zipping across a molecule with attosecond level precision. Okay, so with our X-ray laser, our attosecond X-ray laser, we can make one X-ray pulse. We can also make two X-ray pulses. And for these measurements, we use two X-ray pulses. One of the X-ray pulses is called a pump pulse, and the other is a probe pulse. And we can very carefully control the delay between these two pulses. So we have one attosecond pulse, which is a pump. That corresponds to a whistle from my electrons. It kicks the electrons off. It tells the sheepdog to start moving. Then there's a delay, and then we can shine a probe pulse on the molecule. And the wavelength of the probe pulse is actually so small that I don't just get a view of this full molecule, but I can zoom in on one particular atom, this red oxygen atom in my molecule. And I can ask the question, well, is my dog, is my electron at this red atom on the molecule? And I've drawn a cartoon of a magnifying glass there. Of course, I don't use a magnifying glass. The magnifying glass that I use is this long thing you can see in a picture here. It's called a magnetic bottle electron spectrometer. And it is a special type of spectrometer we can use to help zoom in on this oxygen atom. So we have a pump that kicks off the dynamics. It's a whistle for the sheepdog. We wait some time, then we ask the question with our probe, how close is the sheepdog? How close is the electron to this oxygen atom? And now I'm going to show you some real data. This is real data that we took a few months ago. On the x-axis, I have the delay, where we're mapping out what the sheepdog is doing. And on the y-axis, I have some measure of how likely the electron is to be at the oxygen atom. So y-axis is how likely is my electron to be at the oxygen atom. X-axis is delay. What is the delay between my pump and my probe? And I can control the delay and so map out in time what the sheepdog is doing. And so how do I understand this plot? Well, at the very start, at very small delays, my sheepdog is quite likely to be at the oxygen atom. But I can tell, I have this arrow, I can tell it's moving away because after this delay, my likelihood falls down until we get to something that's not that likely. At this second point here, the second indicated point here, my sheepdog is not that likely to be at the oxygen atom, but I can tell that it's moving towards it because a bit of time later, here, again, it's quite likely that I'm going to measure my sheepdog at the oxygen atom. So I can map out my sheepdog running from red to blue, blue back to red, and then something weird happens because it sort of goes away, okay? It's running one way, it's running the other way, and then we have a lost dog. And actually what we're measuring here is where the really interesting bit starts to happen. Because we have models that can help describe how the dog moves from the red to the blue atom, back to the red atom. Models that describe how the dog is lost, how we no longer see the dog at all, we don't have at our disposal right now. So we can bring this experimental data to our theoretical collaborators, our collaborators who build models of this science, of these molecules, and we can ask them, we can say to them, we have this data, can you refine the model that you have? Here's the data to help justify what we see. And we know a little bit about why this point is low. We know that it's because the molecules, the nuclei, these balls in the molecules, have started moving a tiny bit. And so one way of thinking about it is the dog has sort of got lost among the sheep somewhere, but we don't know where, we don't know how. But we know that the nuclei have just started moving, and that means that our dog has become a little lost. Okay, so we can watch an electron zip back and forth across the molecule on a scale of attoseconds, extraordinarily short. And this is a new experimental capability. This isn't something that we were able to do before. What can we do with it? Well, I said at the start, electrons make things happen. If we can catch electrons in action, we can watch electrons move across electronic circuits, we can watch them carry solar energy in solar cells, and we can watch how they initiate chemical reactions. What's next is that we hope that our understanding, our first measurements of how electrons move in big systems can help us design the next generation of microelectronics, the next generation of solar panels, and even new catalysts for applications such as clean energy technology. And moreover, if we understand how electrons move and we can watch them move in molecules, we can even see what happens when light initiates changes in systems. Why, for example, is photosynthesis so much more efficient than our best solar panels? Why does DNA get damaged in particular ways when sunlight shines on it? And if you're so inclined, good luck finding research funding. Why does champagne start tasting bad when sunlight shines on top of it? And the last thing I'm going to speak about is something which is very exciting, and it's happening right here. I've spoken about all the experiments we did with this long linear accelerator that generates electrons that move very fast, that generate X-ray pulses that then let us look at electrons again. All of the experiments I've spoken about initially, up to now, were made with an accelerator that was built in the 60s. It was refurbished, it was reused, but it was built in the 1960s. And it generated 120 pulses of X-rays per second. Very recently, we completed an upgrade to what we're inventively calling the LCLS-2. And this LCLS-2 takes this first third of the accelerator, introduces new superconducting accelerating cavities into it, and this will allow us to generate not 120 pulses per second, but 1 million pulses per second, an enormous increase in the number of pulses we can make. And if we can make more pulses, well, we can do more experiments, we can find out more in the same amount of time that it took us to do a fraction of the number of experiments before. And it's exciting that the first experiments on using this new ultra speedy accelerator will start next year. Okay, so I'm going to end by saying, of course, this work was not done alone. It was done with a huge number of different people making very, very important contributions. This is the group of people, the core group of people I work with who focus on attosecond experiments, but there are a huge number of other people who also help make these experiments possible. And that is my last slide. I will finish with a picture of Flash and say thank you very much, and I will be delighted to answer any questions you have.
Okay, so Taran, thank you very much. We do have time for some questions now. So we'll run the questions in the following way. Please raise your hand to be recognized. Then these kind people here will run up and down the aisle and hand you a microphone. And when you get the microphone, then you can ask your question. This lecture is being recorded, so your question won't be on the recording until you have the microphone. So, sir, why don't you start?
Yeah, hi, thanks. What does it mean electron is more likely to be here than there? Does that mean it's going to react or has more charge, or what does that actually mean?
Okay, so that's a great question. So we can describe electrons as waves. Okay, any experiment that we do in the world, we can devise a model that describes those electrons as waves. And if we describe an electron as a wave, well, when we look at an electron, so-called electron, we can do a measurement like I spoke about, which asks, like, where is that electron in space? Say I have some box, and I look at the box, and I want to know where that electron is in space. Is it here? Is it here? Is it here? Is it here? And we know from experimental measurements that when we look at an electron, it actually is in one of those positions, right? We don't see a wave, we see an electron in one position. And you know, Einstein has this famous quote, God doesn't play dice. Yeah, that it somewhat relates to this question, right? Because it turns out that when you somewhat relates to this question, it turns out that you can describe completely a system using a wave, but something weird happens when you look at that system and you don't observe a wave, you observe an electron in one particular place. This is a very fundamental problem with quantum mechanics, that everything behaves like a wave until you look at it, and then it behaves like a particle. It stops behaving as much like a wave. And so when I say like how likely an electron is to be there, you could think of me having a very rigorous mathematical model that describes everything about an electron moving in, say, a box, which describes an electron as a wave. And then I look at that electron and suddenly it becomes a particle. And I can't know everything about that system. I can only know where it's more likely to be. And this is relates to like God playing dice. It turns out that you can never say for sure when you look at an electron where it's going to be. You can only say where it's more likely to be. Does that make sense? Cool.
Okay. Next, please. Sir, up there. Okay, let's let this fellow go first, then you.
Oh, thank you. You mentioned your lab was underground. Is that intentional to provide some kind of isolation, or was that just a convenient place where all the stuff ended up?
Definitely wasn't convenient. Yeah, you got it right, exactly. The reason the lab is underground, well, actually, I've worked in a number of different laser labs with lasers of different size. The smallest lasers you can get like sit on a tabletop. They're still very sensitive to vibrations. So typically we try and build any laser lab underground because that mitigates the effect of vibrations. Like a car driving overhead can change it. The LCLS and the SLAC accelerator is like an extra additional piece of that, right? It was built all underground to protect from external environmental perturbations. Missing the fact, I think it was built across the fault line. Is that right? Or very close to a fault line? So there's
up to the fault line.
Up to a fault line. Okay. So, but yeah, it's exactly that. It was the accelerator system was built underground. It's quite hard to bend X-rays. So the easiest thing for us to do was to tunnel down and build a lab that accepted X-rays from the accelerator also underground. Yeah.
Okay, sir.
Hi, thank you. What temperature pressure regime are you taking your measurements, and how are you fixing your organic molecules so that they don't just zip away while you're taking the picture? Are they fixed?
Yeah, okay, great question. So you asked the pressure. So we run this, I do gas phase experiments, okay? And you put things in the gas phase because you can, if you see something interesting, right? If you're measuring a liquid or a solid, you're not sure if it's because one molecule is interacting with a molecule nearby in a liquid or because it's an isolated molecule. So basically, it makes experiments easier to do if you just have something in the gas phase and you look at a particular molecule. And the pressures we run at are on the order of like 10 to the minus 7 torr, something like that and below for the pressure of gas phase samples. Regarding molecules being fixed in space, what happens here is they're not fixed in space. They shoot what I have as like a needle that I have a so-called oven, which is a little temperature reservoir built by Eric, who's running a mic there. And you heat up this little reservoir. It causes molecules to shoot out of a needle. And so they're spraying all over the place all the time. The thing is, though, they're moving fast, but not that fast. So if I have two pulses, which are separated by a femtosecond or a few hundred attoseconds, well, actually, the molecule hasn't really had any time to do anything except be in exactly the same position between when the two pulses were shot out. There are things you can do to fix a molecule in space. You can use other laser pulses to do that. It's a great question, but for what we're doing, we look at dynamics that are so fast that basically the molecule is fixed in space anyway.
Let's start here and then.
You said you'll be able to produce 1 million pulses per second. What's the duration of a pulse?
Great question. So I guess there's two types of pulses we can speak about. There's electron pulses and light pulses. The light pulses, which are the ones that I guess I'm most interested in and we use, will be the same duration as the ones we spoke about. So we made, with the previous X-ray laser, the shortest pulses we could make were about 200 attoseconds long, and we'll be able to do exactly the same thing with the new accelerator. So actually going from the old to the new accelerator, we'll be able to do all of the good things that we could do before. What's nice is that the new accelerator will be a bit more stable as well. So our pulse energy and our, for example, the photon energy, the wavelength of the light that you generate has more stability than it used to be before. So yeah, my answer is the pulses will be the same length as the previous accelerator, and the shortest ones we can make are right now about 200 attoseconds long. You can also make them femtoseconds long as well.
Right, and then between pulses, you get a whole microsecond. It's a huge amount of time. Over here.
So it was great to hear that in 2023 and next year, most probably, you will be able to take a picture of the electron in an analogy, right? So, and this will mean understanding this box, which we didn't understand yet. Now I'm thinking, so it will have a lot of implications, as you mentioned, into micro electromechanical systems like the semiconductors. So do you have any projections or like any hypotheses? What will it change, the technology that we can use, or like any, it can be an open question, I know.
It's a great question. Yeah, it's a great question. And obviously, So I guess your question is, what will the ultimate technical advances be that we can produce from the science? Yeah, okay.
Curious in advance.
Yeah, great. You sure you're not a research funder sneaking in here and asking a question? Because that's what the research funders will say as well. I guess I would answer you in a couple of different ways. One is that a lot of the science we do is really so-called blue sky science, right? So we do it for the sake of doing science. And because we know from experience that when we understand more about how systems work, we build new technology that we can't necessarily predict, but that we can build. One example that's quite far-fetched is like, the internet came out of CERN. People built high energy. I'm not saying it was, justifies all the cost there, but it was an additional technical advancement that was needed to help manage all the information they generated. And yes, we can. A lot of it is blue sky science. In terms of like more fundamental applications and where in particular we see this sort of work going, one open question is, why is photosynthesis so efficient? So we can't build solar cells which are nearly as efficient as photosynthesis, and we know that electrons are the things that see the light and will help convert the energy into useful electrical energy. A lot of photosynthetic cells have electron dynamics which are in solids, which are much more complex than the systems we study right now. But to understand how electron dynamics unfold in more complex systems, we need to first take ourselves back to systems that we can understand, which are these isolated gas phase molecules I can speak about. So one answer could be with the acknowledgement that I don't know how long it will take, and I also don't know what other obstacles there are on the road, is that the quantum mechanics of electrons does something to make photosynthesis much more efficient, we think. And understanding that in a simple system and expanding that discovery to larger systems will be something that I hope we might be able to understand. We can also, sorry, it's a long-winded answer to a question. I'll say one more thing. We can also, on a very small scale, change how chemical reactions happen right now, okay? So using this knowledge, we can change a laser pulse that we shine on a chemical reaction happening and actually divert the products. So change the products from being one thing to another, which is crazy, right? We can divert the path of a chemical reaction. I'm sure you can think of a bunch of different ways that might be useful. Scaling it up to an industrial level is very hard, but that is another application that will definitely have lots of uses. It's just further technical developments that wait to tell us exactly what.
Okay, there was a question over here.
When you say the location of your electron, there's a whole bunch of electrons in there. So how do you know which one is yours?
Yeah, excellent question. Yeah. Okay, so great question. What happens is, there are a bunch of electrons. Okay, so what happens is the whistle that we use, we know which electron that is addressing, okay? So we know that this whistle is addressing a particular electron. I didn't go much into the explanation of this. It also turns out that X-rays are useful because you can whistle at not just a bunch of sheepdogs in the molecule, but a given sheepdog in the molecule. So we know which electron is moving because we know which electron we whistled at in the first place. And yes, typically the electrons that we might choose to whistle at are so-called valence electrons, the most weakly bound electrons in a system, because the valence electrons are the ones that participate most strongly in these chemical bonds.
Maybe it's worth saying that if you use more energetic X-rays, it can be very element specific. So the X-ray will affect a particular electron in the oxygen atom, but not a comparable electron in the nitrogen atom. And that's another amazing thing about these experiments that you can explore. Okay, this lady has one question.
What do you think would happen if two of these electrons in the thing would like smash into each other? What would happen?
Yeah, great question. So what would happen if these two of these sheepdogs smashed into each other? Okay, so what the answer is that happens all the time. They're interacting with each other all the time. And I spoke about at the start, do you remember at one of the very early slides I had, I said, we don't know very well how electrons, the sheepdogs, speak to sheep. That's a really hard question to answer. We also really have a bad understanding of how these different sheepdogs speak to each other. When we have more than a few sheepdogs interacting, that problem is also really hard to solve. And that's also a really important problem because, say, for example, in your phone, okay, if you have a phone with electrical energy moving around, there's not just one electron, there's not just one sheepdog moving in there, there's a huge number of different ones. So my answer is, we have some models that explain the fundamentals of how electrons bump into each other. The most simple answer I can give you is they repel each other because they have a negative charge and they repel each other. But beyond that, it's a great question, and might be something you want to consider studying because it won't be solved by the time you get to college.
Okay. Somebody up here have a question? Yeah, the fellow in the back row there.
I was wondering about, you mentioned there's a pump and then a probe. Is there a time varying element to the state of the electrons prior to the pump? And if so, then is the pump putting it into a consistent state, so you're measuring the same thing, or is this like a statistical average of like possibly different states? I'm just wondering like what is, and
Yeah.
especially if like large molecules, do they have a varying state over time or?
Yeah. Great question. So you're asking like, is something in motion before we come in with the pump? And the answer, okay, so we do a few things to make sure that that's not the case. Okay, so for example, we have an effusive needle, which makes the molecules that we have at the interaction region very cold. We intentionally make them very cold at the interaction region. And on a molecular scale, something being cold corresponds to the nuclei, at least, like not moving at all. And in quantum mechanical terms, the electrons themselves, these sheepdogs, are in their so-called ground state, okay? And what that ground state means is it's the lowest energy configuration they can get to. So really, there's no quantum mechanical motion that's happening. So yeah, it's a great question. We have to think about making sure that they're in the ground state, both the electrons, the sheepdogs, and the nuclei are in their ground state. But for the experiments I've spoken about here, we can pretty safely assume that yes, they are. There's nothing happening before we come in and blast them.
Okay, so we'll take two more questions. And the first one goes to the fellow in the front row.
Thank you so much for the talk tonight. This could possibly stem off of what you were just getting into about the electron being in a ground state. And this might also be a laughably silly question, depending on what the final answer, I suppose, might be. But since particles travel different in different mediums, is there any thought to infusing the accelerator with something like a stable gas or just something that allows an easier measurement that would allow kind of a controlled slowing to better measure, assuming you can control for the effects of that medium, or does that only muddy the observation field?
You're talking about, okay, yeah, great question, cool. You're talking about taking this thing, right, and putting it in something that slows the electrons down. Is that right? Yeah.
Yeah,
okay.
That's right.
Great question. So you can do that. Problem is, we know that maybe to say electrons, we know that electrons, yeah, sure, they can get slowed by being in like a liquid phase or a solid phase. We know that. How do they get slowed? Very hard to answer. So in fact, it's a great question. It's like a payoff, okay? It also turns out an interesting point is that the smaller the molecule you get, the faster electrons move. Electrons actually move a bit slower in bigger, floppier molecules. So we can do things to make the electrons move more slowly. We can, for example, take this and pile it up into a polymer of 10 different units of this. But the payoff that we get is that when we have a molecule 10 times bigger, there is a vastly nonlinear increase in how hard it is to describe it with our models. So we can slow them down, but that just makes things even harder to understand. So one main reason we have to go quite so fast is because we need to study really small molecules, because those are the only ones that our computational models can properly understand.
And that's more about the measurement at the moment, not just the behavior becoming erratic.
Right. Yeah, exactly. Yeah. Great question.
Okay, last question, this fellow here.
So around the middle of your talk, you made the statement that we can deal with the quantum mechanical issues because we understand the wave mechanics. And you presented 4 component waves leading up to a total wave. Was that just kind of a theoretical statement, or is there some actual evidence for those component waves that we have? And if not, is your work going to contribute to that?
Oh yeah, okay, great question. So yeah, that is it is much more than a theoretical statement. We can, yeah, okay. So electrons in many quantum systems we had to describe as waves. And to give you an example of how this comes out. So electrons are hard to look at, right? They're hard, they're small, they move fast. These molecules, these ball and sticks, are actually a bit easier. They move more slowly and they're bigger. So we can look at those structures. And the electrons, like I said, decide where the sheep sit. And this is where the nuclei sit. And this is also true for the stationary case, right? So if I take a bunch of atoms, crash them together, they will eventually settle in one configuration and not another. There is a reason this molecule has this shape and, this isn't up off there somewhere. Using the wave model of quantum mechanics using the model where we describe these electrons by different waves, we solve the Schrodinger equation to get the shapes of these waves. We can predict how these nuclei will arrange themselves, and we can measure those. And when we measure how the nuclei have arranged themselves, it's phenomenal accuracy that we can get and have been able to do so for many decades by using this concept of electrons being described by lots of different waves adding together. So yeah, it really is a model. I drew a toy model, but that the overall theoretical framework is really something that's been extraordinarily rigorously tested and shown to be true in a great many different ways. Yeah.
So thank you all very much, and let's thank Taran once again. Okay, so let me just say, we'll hang around here for a while in case you want to ask more questions informally. But I think the people who maintain this building would like to clean up and finish with the audio here. So let's all move out into the foyer, and Taran will be there and a couple of his colleagues, as you see, to answer the questions that you might have. And in general, thank you for coming. The next of these public lectures will be at the end of January, so please look for the announcement. And if you don't get the mailings, just go to the SLAC website, look for public lectures, and get yourself on the mailing list. So again, thank you all very much.